All right, for the last part of Project 1, you're going to need to be able to compare your sequence to another organism. So, pretty straightforward here. Um, but you need to go download something. I would suggest, actually, I'm going to restrict you no apes. In other words, you're going to compare your version 2. So, first, let's do that. Let's find our GPX1 uh, variant 2. GPX1 Homo sapiens. Let's do that. And, it'd be help if I could spell. You can just put in human. It knows what human is. There we go. All right, and I'm going to do, let's look for variant 2. That was the long one, so I'm going to go grab that FASTA here. Um, and I'm just going to grab it like this. I'm just going to grab the sequence, right click and copy. And what I'm going to do is we're going to go to BLAST. Now, if you don't remember BLAST, we did a worksheet on it. Here it is. You can instead go NCBI, search in Google NCBI BLAST in, and that should bring you to this page. But you're looking for BLAST in, and what I'm going to do is I'm going to put in my sequence like this. Or, actually, it says you can put in your accession numbers or your GIs. So technically, you really don't have to even put in your sequence. Let's see here. You could just put in this number up here. In fact, why don't we do that? Let's just do it this way. You could do the whole sequence, or you could put in that little number there that comes in here, and you can put it in here. That's all you actually have to do. All right, now what I want you to do here is I want you to compare it to another organism and avoid apes. That's too close. You're either not going to get anything different, or you're not going to have anything to talk about. Could be a little too easy, or there's just nothing to talk about because it's identical or it's too close, so that it wouldn't be interesting. So too far away would be something like a plant, so you don't want to compare it to a plant, or certainly not to a bacteria. So somewhere in between, mice, cats, cows. I'm going to try a cow, just because they always do something interesting. Cows are kind of interesting in that their genes have some interesting things that go on. If you didn't know, cows, for the most part, don't get cancer. So people study cows just for that reason. So let's just see. I think it's boss something, but let's just see if it's smart enough to know cows. And so, nope. Let's see. What I want to do is I want to restrict it to a comparison to a specific organism. So you can exclude them or add them. Let's see. Boss. Let's see if I can remember it. Boss bovis. Okay, I think this is it right here. So I'm going to do cows. If it's not cows, it's going to be something close. A moose or something. So here we go. We're going to do this organism where I'm going to compare our sequence to the entire genome of this organism. I want you to pick one. Find something that is not a plant and not an ape. Some animal. Actually, one of the more interesting ones are reptiles, frogs, amphibians, because they're far enough away to get some interesting things. But, a cat, a rat, all of those are potential answers. So what I want you to do is do that and leave it in the standard settings at first. And if you don't find anything interesting like I hope I find here, you can change these if you want to stick with your organism. If you don't find anything interesting in the organism that you understand, change it to another organism until you find something that you would like to talk about, something that makes sense to you. I don't want, this is more of a fun, just kind of explore kind of exercise here. But let me show you what I want you to be able to tell me. In other words, I want you to understand how to use BLAST and what it can show you. So watch this as I search, and let's see what comes up with cows. So we wait just a second here. Let me make this full screen. Sometimes it can take a while, and it's found nothing similar. Now that's interesting. If you get that, that means, well, you can redo the exercise with a lower setting, or you can switch to another organism. So obviously cows are different in GPX1. So let's go back and let's change this to somewhat similar, and I'll give it one more shot for cows. Otherwise we're going to switch to rats. Now whatever I do it on, you can't do it on. So if I do rats, you can't do rats. Yeah, waiting. Here we go. So when I lowered the frequencies, I actually found something. So what did it tell me? Well, it gave me Homo sapiens. I don't want that. Did I unclick the cow? I must have. So don't do that. Yep, I sure did. When I redid it, I didn't put in. So let's go ahead and put back in Bos Taurus and Bos Bovis. Bos Taurus is, I think, actually cows. So who knows what we're actually studying, searching for here. I'm just doing this as an example to show you, you know, what you could get. The whole idea here is I want you to kind of explore with this, find something interesting that you're interested in, and do a BLAST search on it. I'm going to do somewhat similar this time, and hopefully we get something versus the other time where we didn't get anything. All right. I always like doing, there are certain organisms I like to, if I'm doing something like this, I like to do against because they usually turn up something that you're not expecting, and cows are one of them. Aha. All right, so what did we find? We found a place on chromosome 22, all right, and we did. We found also, I suspect that their GPX1 is on their chromosome 22. So here's the one I'm interested in. So let's click on this one and let's look. So what it shows here is, let's see, we want not 22, which one? I should have stayed where I was. mRNA, no, yeah, this one here. So it shows you that there are two spots on the cow. So how you do this is you scroll down. When I clicked on it, it went to this one. So I clicked on this and it sent me down here. So what I want you to do is download this as a FASTA. So the complete sequence. All right. Now you can then compare it. Now the other thing, actually, you know, it might be better to download that as aligned sequences. So not complete, do the aligned sequences. It'll give you something closer to this. Here, let's go ahead and do that. I'm going to continue. All right, and it's going to give you something like this. And this is basically the two sequences in an aligned fashion. And it shows you the two FASTAs right there next to each other. And these are the pieces that are the most aligned. Now, you don't see it. What I want you to do is to compare it. Now, you have the two sequences when you do it this way, but that's not really what I want you to turn in. What I want you to turn in is something closer to this. And I want you to notice, all right, in your sequence, what changed. Mainly, look through these two parts. So there are two pieces here. It breaks it down into two pieces and says, this one and this one aligned. You'll have to figure out which one is cow and which one is human. All right, and then you look to see what changed. Here, obviously, something got cut out. Some pieces got cut out towards the end. Here, some pieces in the middle. And then, of course, the fact that it breaks it down. Notice on one of them, you go from 908, 350, and this one starts. So there's a big chunk cut out of these. So in other words, notice that this one stops at 350 and starts back up. Well, that is 350. So it is pretty continuous. Apparently, the algorithm they have, the computer program they have, distinguishes something different in that area and decides to break it up to 350. Let's see, 605. But this one is only 328. So there's your difference. All right. So what you need to do is compare these, figure out what's going on. The subject here is 348, and it aligns with right in here, GG. Let's see, is that a GG? Two GGs. Yep, right there. A before it. Yep. So this AGG right here is this AGG right here, and this AGG here is up here, and that one. Okay. So this is telling you that this is actually very interesting. So we did find something interesting. Essentially, there's a piece of this sequence that has, let's see, which one had the duplication? There's been a duplication of the sequence probably. In other words, this 350 at the bottom is the same as the well, no, this is before it. This is before it. So this one starts here, 350 lines with 350, but here it aligns with 664. So there's a big chunk missing out of variant 2. So if you can figure out what I'm talking about here, in other words, why did it break it up? All right. Realize, of course, one is longer than the other. All right. And the best way to do this, if you can't figure out what's going on between these two, is to actually take these two and do a dot plot. This is why I always tell you dot plot is your best friend. So you would take these two sequences and run them on a dot plot, and that will visually tell you what was happening between these two. Now, beyond that, on this part, what do I want you to do? Well, if you do a dot plot, which you probably should no matter what you do, go ahead and put that dot plot in the answer and explain what that dot plot is telling you, if anything at all. And then look at these pieces and look to see how they're aligned. How close or different are they? Well, identities, 84%. So out of these here, 84%, and up here, 81%. So they're close, but they're not really that close. There are quite a few mutations in here. And what you would tell me is that, hey, there are lots of places where there are just places that are missing. Look at this, here and here. Now, in a later analysis, when we move on to the later part of the class, we would go in and we are going to grab that sequence there, and we are going to look up and see if that sequence that was added or taken away has particular function in biology. Now, we're not going to do that one on this one. On this one, all I want you to do is notice things like that. There are a couple of sequences here that are missing. Are these perhaps things that might affect a splice site? That's something. Go look and see if the letters are something that has to do with the splicing. Now I know what this one does. This is actually part of this. This implies that there was a transposable element, something called a jumping gene, that jumped into this sequence and altered it, which is really interesting to me. I did not know that. This one here is the same thing. It could also indicate the splicing has been added, which makes complete sense. That's one way genes evolve, is that they gain or lose splice sites. So look to see if the splicing rules, if any of the sequences in these pieces, towards the ends or something. But also give me a dot plot. Now this is not the only thing I want you to do for this part. We also want to. This is alignments here that I'm on. You could also do a graphical summary. This is showing the pieces that that stayed the same. This is just the first one. We're just interested in one, but it is interesting. This is kind of giving you the graphic, something that the dot plot would give you. It's kind of showing you where pieces might be cut out, which is right here. The dot plot will do the same. This is your description, your alignments. Now the taxonomy, you don't have to do the taxonomy. Now let's see, there is something else they've taken it out. They usually give you a little dot plot whenever you do alignments. I don't see it. They must have taken that feature out. They used to give you, it was a really bad dot plot that gave you no real information, and it was really hard to use. So I guess they finally decided to just take that out. But often you can get the you used to be able to get a dot plot whenever you did alignments, and there's no dot plot here. Let's see, maybe if I go to the one we were doing there. Let me scroll back down to it. Is this it? This is the one, yeah. Let's see if I go to graphics right here for this particular piece. If it'll give me the, no, it's, let's see what it's going to give me here. Oh, I know what it's going to give me. We haven't gotten to this part yet. This is for later in the course. This is closer to what Eugene does, but I don't see dot plots here, so you're going to have to do your dot plot on your own. Yeah. But notice over here you get lots of interesting things you can learn about GPX genes. I don't think tools, blast, new panel. Nope, no dot plots. All right, so you can't do that. But notice that the E values, you can report them if you like. I'm not really looking for E values on these. The percentage change is what I'm looking for. And I want your interpretation. Look at what you get. So on this one, we were looking at this, and we see these pieces cut out. At this point, what you would do is look for things that are odd, all right? Now this is still the same. These are mismatches. See, there's some places missing, and the sequence is a little different. You would notice these. Just if you want to, you can do like this and copy this piece and put it into the into your report, and then talk about this whole piece, because this is the interesting piece right here. And this one, there might be a couple of interesting pieces here, and both here and here. Here are some mismatches, things that don't, go together, don't align. Let's see, some interesting things. So look through here. Now it would be also interesting to find out, all right, if where the start and stop codons are and the splice sites. If you've done the previous part of your sequence, you can find those here in your sequence, which I suppose is the top one here, the long one, 1000, yeah, the top one. So you would go through and find your start and stop codon. Probably it's, let's see, ATG right here probably. All right, that looks to have stayed the same. So report that, and let's see, stop. You're not going to be able to, it's going to be more difficult to find the stop, but that would be interesting to see if those are still the same. You could look to see if the splicing, wherever you think the splicing was before, if the splicing is similar here. But you can see that the sequence has enough similarity that you can tell it's the same gene. Look at how similar. I mean, the fact that it's 81% in this part, 84% here up in the beginning of it. But there are some major differences, and we want to just I'm just trying to have you focus on here's how you would find the differences between two organisms. Why are their genes different? Right here. These are the changes between a cow, or this is something similar to a cow. I'm not sure it's the exact cow. I'll have to look up and see what Boss. Boss Taurus. That's a cow. Yeah. The differences in a cow here. You expect differences in cows because of certain things, and actually, this would make sense that it's maybe a transposon because they handle transposons different, and it might be why they don't get as much cancer. They don't get cancer at all, really. Anyway, so the idea here is check this out and just notice the differences. So give me give me the picture of this. You can copy this. All right, copy the whole thing. Focus in on some. Maybe you just focus in on a couple of pieces. Like on this one, I would focus maybe on this one, and here, and then you just report, hey, I noticed this interesting change. And if you have time and want to research it, you could grab some of these sequences and search just on Wikipedia. Don't go overboard, and see what maybe some of these things were for that are missing. This piece is missing in humans, and this piece is missing in cows. Does that make a difference? Then you could look down here and check out maybe this sequence. It looks kind of interesting. And go search, just a simple search. And if it's something that's out there that's commonly known, it should show up and you can report that. I don't expect you to go into detail. You just need to pick out the main features. All right, the main features that are different between these two. Here's an AG that's missing. Not a big deal. It is a big deal, but it's here you have maybe an insertion or deletion plus a there are different places along here, but these larger pieces are probably the more interesting ones. All right, here's an interesting one where you have two on either side. All right, notice it's a TG and a TG. That's probably some kind of duplication, but like I said, just these are things, just notice where the differences are and comment on them. If you can find something on Wikipedia or something when you search for this. So let's just do that. I'm going to take this, and I'm going to take this piece that got cut here, copy, and why don't we just go search, and we'll say molecular biology. Why don't we kind of give it I'm just going to paste. Okay. And let's see, or you could put in bioinformatics or something like that and see. Holiday Junction. I thought so. I thought I recognized something. So maybe this is what. And oh yeah, here we go. For topoisomerase. Interesting. Okay. So there you go. We found something. All right, and you could just report on that. I was not expecting to find anything here, and actually it really was a piece. I thought it was probably part of a transposon, but it looks like it was a virus infected this and probably got spliced into the cow. The cow got a cold, and part of the virus ended up getting spliced into its genome. Makes sense. All right, so see, found something interesting right here. I'm not requiring you to find something interesting, but you can report on whatever you do find. I knew cows are interesting, and there are usually things to find in cows. So the idea here is to just play with this last one. Give me whatever your results are here. All right, for the one that matches your organism. Now, it might be kind of like, why did I skip these first two? Because those are boss torus. Those are assemblies and the whole chromosome. Don't want the whole chromosome. I wanted to compare it to an mRNA. So you notice I went to the mRNA. So keep that in mind. Look for the mRNA. Don't choose the chromosome. If you don't get an mRNA, switch to a new organism or something. But compare it with your version 2 and use BLAST to do that. Report what you find in here. However you want to make this visual to me, that's fine. I do like this type of visual here, and you probably ought to include a dot plot of your comparison. So in other words, of the variant 2 in humans to the to this one, which is the Bos Taurus mRNA for GPX1. Does it give a variant? Bos Taurus. No, it does not give a variant. They probably haven't researched it enough to find the variant, the splicing yet. So that's the advantage in humans. We've done a lot more research. So we have variants that we know of the splicing, and they probably haven't done it. They just give the only mRNA they found. They may have only haven't put in the time to find all the different RNAs. They probably just found the one that was the most commonly, the highest percentage present in the cell. Probably haven't done different cell types or anything like that. Realize a lot of these spliced variants only occur in some cells in the body. So like GPX8 only occurs in GPX, yes, the entire gene GPX8 only occurs in the neuron, but also some splicing only occurs in some cells as well. So just like you have GPX1, 2, 3, 4, 5, 6, 7, 8, and then each one of those is going to have a variant based on the splicing. Well, you may have cells that only have GPX1, but they also may only produce one variant, not multiple variants. So keep that in mind. That's just something to know. And so you usually, if you're sequencing, you can't just sequence a lot. Or if you're doing mRNA extraction, you might not be able to get away with just one cell type if you want to know everything that's going on and all the splicing. You might have to try several cell types, and as you sequence or you try to gather mRNAs, you might have to go through many tissues, and we haven't even gone through all the tissues with all the genes in humans yet, so much less a cow. I'm not surprised. All right, so I think this is straightforward. If not, this is actually the last required part of the Project 1, and I'm going to do another video on the extra credit, and I'll also do another one to kind of end it, to kind of put it all together and show you the formatting. We're not going to do a quiz this week. Instead, I will let you work on the project to turn it in. All right, and that's it for today.